This is the Drug Delivery Lab, and we are Group 10, Gina Ducati, Morgan Trefny, and Keely Walters. The objective of this experiment is to determine which conditions lead to the greatest fraction of dye being transferred from hydrogel beads to the body, along with the rate of drug release for each condition. In order to do this, we must analyze the mass transport of tartrazine, which occurs from the hydrogel beads diffusing into the deionized water. This experiment models drug release from delivery systems controlled by diffusional mass transport. Drug delivery is the process of administering a pharmaceutical compound to achieve a therapeutic effect. Controlled drug delivery systems administer a drug at a predetermined rate. It is imperative for the drug to be released at this rate to achieve maximum efficiency. By doing this experiment, we are able to mimic a drug entering the body and determine the optimal condition for drug delivery based on the Fickian diffusion mechanism. The conditions we chose to vary for this experiment are the mixing speed, temperature, and bead size. By measuring the absorbance of the solution comprised of water with the tartrazine hydrogel beads over time, it is possible to determine the rate control mechanism constant and the fraction of dye release. To perform this experiment, we needed to research the theory behind drug delivery. The rate control mechanism constant, or N, defines the type of mass transport across a surface. We can define the mechanism of diffusion in two ways: Fickian and non-Fickian. The type of drug release mechanism is Fickian if it has a numerical value less than 0.43, and is non-Fickian if it has a numerical value between 0.43 and 0.85. This exponential equation shown above describes the general solute release behavior of a controlled system for the first 60% of drug release, regardless of geometric shape. Mt corresponds to the mass of the drug released at time t, while M infinity corresponds to the mass of drug released at time infinity. By performing the experiment and acquiring absorbance readings of a solution over time, we can generate values for the dye release, or F, and solve for the kinetic constant and the diffusion rate control constant. As previously stated, we can solve for the kinetic constant using the equation on the previous slide. However, to do so, we must manipulate the equation to mimic a linear model. The final manipulated equation is expressed above, as the ln of f equals n times the ln of t plus ln of k. By plotting the natural log of f and the natural log of t, we can use the slope of the linear trend line to obtain n and then k. Also, to observe the rate of release within the system, the derivative of f must be taken. The mathematical equation for rate of release is equal to the change in f over the change in t, or the slope of 2 points.
The general procedure of this lab was to observe and determine optimal conditions for drug release. In order to do this, we varied 3 different factors and took absorbance readings using the peak absorbance of tartrazine, which was 427 nanometers. A couple of safety precautions that we had to take were to not pour alginate down the sink and to wear appropriate lab attire because calcium chloride is a skin irritant. The first step of this experiment was to produce the solutions that we would be using throughout the lab. There were 3 different solutions that we had to prepare for this experiment. The first was a 1-liter solution that was 0.5 grams per liter tartrazine. We made this by measuring out 0.5 grams of tartrazine and adding it to 1 liter of water. The second solution was the alginate solution. In order to make this, we added 0.1 grams of alginate per 10 milliliters of the 0.5 grams per liter tartrazine solution that was made as previously shown. We added a total of 6 grams of alginate into 600 milliliters of the 0.5 tartrazine solution. We let this stir vigorously overnight until a smooth, uniform yellow solution was formed. The third solution we made was a stock solution of 6 weight % calcium chloride. We made this by adding 42.28 grams of calcium chloride dihydrate to 500 milliliters of water. The second step of this experiment was to calibrate the spectrophotometer. We set the wavelength to 427 nanometers, zeroed it out using DI water, and took a couple readings from the known tartrazine concentration to create a calibration curve. The third step of this experiment was the bead production. We poured the calcium chloride solution into a large weigh boat, filled a syringe with 3 milliliters of alginate, and slowly dispensed the alginate into the calcium chloride solution, creating the beads. We let the beads cross-link for 3 minutes and then separated them from the calcium chloride solution by filtration. We rinsed the beads with DI water and then poured them into a beaker filled with 100 milliliters of DI water. This beaker was on a hot plate at a set temperature and mixing speed. The fourth step of this experiment was the measurement of dye released. In order to do this, we removed samples with a pipette and put it into a cuvette, and then placed it into the spectrophotometer and ran the solution, receiving the absorbance. Once the reading was taken, we poured the sample back into the solution. Absorbance measurements were taken every 2 minutes for 10 minutes, then every 5 minutes for 20 minutes, and then every 10 minutes for 30 minutes, equaling a total of 12 readings in 60 minutes. For this experiment, we chose to vary the mixing speed, bead size, and temperature conditions. Displayed is the average absorbance reading for 3 trials when setting the mixing speed to 100, 200, and 300 RPM. The error bars were made based on the standard deviation of the average absorbance readings for the 3 trials. The 200 RPM absorbance reading plot has large error bars due to significant variation between the 3 trials. This graph shows the average absorbance readings of 3 trials when using 0.7 mm, 1.1 mm, and 1.6 mm beads. Once again, the error bars are based on the standard deviation of the average absorbance readings for 3 trials. This graph shows the average absorbance readings for 3 trials when setting the hot plate to a temperature of 25 degrees Celsius, 50 degrees Celsius, and 75 degrees Celsius. The error bars are made based on the standard deviation of the average absorbance readings for the 3 trials. Based on the absorbance readings, we could compute the fraction of dye release at each time point. Using this, we can graph the rate of dye released over a time period. Using the absorbance readings, we can compute the fraction of dye release based on the empirical equation for drug release. By manipulating the empirical equation to become the natural log of F equals the diffusional exponent times the natural log of time plus the natural log of K, we can plot ln F versus ln T and use a line of best fit to determine the diffusional exponent and the kinetic constant. In this plot, the slope is 0.408, which would be the diffusional exponent, and the y-intercept, negative 1.2873, would be the natural log of K, giving us a K value of 1.508. We only graph the first 60% of release behavior in order to accurately measure N and K. Based on the diffusional exponent, we can determine which type of drug release mechanism it is. A diffusional exponent below 0.43 tells us that the drug release mechanism is Fickian diffusion. A diffusional exponent between 0.43 and 0.85 is a non-Fickian transport. All but one of the conditions are Fickian diffusion. The condition with 300 RPM, 25 degrees Celsius, and 1.1 mm beads is non-Fickian due to its 0.45 diffusional exponent. Overall, this experiment allowed us to model the rate a drug is metabolized in the body. Based on our results, we found that increasing the mixing speed led to an increase in the diffusional exponent and an increase in the kinetic constant. We did not find conclusive results by increasing the bead size. This could be due to experimental error. We also found that increasing the temperature led to a decrease in the diffusional exponent and a decrease in the kinetic constant.
Thanks for listening!